Hello and welcome to biology class. Children, today we are going to begin with last lesson of the syllabus, that is skeleton and movements. As you know, this particular lesson, it totally includes a study about movement and locomotion. Movement. Movement vary from different organisms to different ways. Everyone knows that many of the organisms, they move from one place to another. Like in unicellular organisms, you may find there are protoplasmic movements, and in some cases, due to peristalsis, the movement occurs, or even in some cases, walking or running. The movement are of different types. But all these are not called as locomotion. So, what exactly the movement is? What is the difference between locomotion and movement? Movement may or may not end up into locomotion or displacement of organisms. Locomotion is actually a voluntary movement which is under our control, resulting in a change in location. But movements are different many a times, like engulfing the food, swallowing the food that passes through the esophagus due to the movement called as peristalsis. Here we call it as a movement and not as locomotion. So it varies in various examples. So today we are going to study about the differences between what exactly movement and locomotion are. Now on our slide we have different locomotion, different movements. Locomotion and movement, they are interlinked with each other. Certain examples are given over here. In paramecium, cilia help to move food through cytopharynx and for locomotion. So based on different types of movements in human as well as in various other organisms, you may find amoeboid movement as one of the movement, ciliary movement, muscular movement, and along with that one more movement is being added, that is whirling movement or movement of flagella using flagella, and hence we call it as flagellar movement also. So paramecium is an example where ciliary movement takes place. Hydra is an example which uses tentacles to capture prey and for its locomotion. Limbs help to change body postures and for locomotion. So various examples we have on our slide with the picture shows different movements in different kinds of organisms. Now we are going to have a detail about what exactly amoeboid movement is. Now, when we talk about locomotion, when we talk about movement many times in muscles you find that involuntary movements occurs, peristaltic movement occurs like in alimentary canal, dilation of blood vessels. Even in cardiac muscles, heart contracts and relaxes. So the muscles over there work voluntary, involuntary in various other cases, like in limbs and all, it moves voluntary and gives the various movement. But when we talk about various movement, we could find here locomotory movements found throughout the animal kingdom. Among them, first is amoeboid movement. Amoeboid movement, here you'll find pseudopodia formed by streaming of protoplasm. So usually pseudopodia, which is a false feet of amoeba. Like various examples are there, leukocyte is also an example where you will find amoeboid movement is seen. So the way extending pseudopodium is seen, there the protoplasm will move and will change the shape continuously every now and then, and hence such movement is called as amoeboid movement. Here cytoskeletal elements like microfilaments also help for this, for the movement. So example are given about macrophages and leukocytes. So this is one of the example, one of the movement we call as amoeboid movement. So as we are talking about movement or locomotion, locomotion is actually movement from one place to another. Maybe in search of food, maybe in search of shelter, maybe in search of breeding ground or escape from the enemy. For various other purposes, the movement do take place. Second movement we are talking here about is ciliary movement. So by the help of cilia, performed by cilia, ciliated epithelium. Ciliated epithelium we have studied in one of the lessons, where epithelial cells have various hair-like structures which are called as cilia, and we could see over here in the picture. Not only these, even in paramecium, cilia help in passage of food through cytopharynx. So this also example comes under ciliary movement. Types of movement in human, like muscular movement, which is one of the movement where muscles with the help of bones and joints perform movement. So we could see here this takes place by the muscles. Example, movement of limbs, movement of tongue, jaw, etc. all comes under muscular movement. Lastly, we have flagellar movement or whirling movement as given in the text. This is actually, this movement helps in swimming of spermatozoa, yes, sperms, maintenance of water current in the canal system of sponges, and locomotion of protozoans like euglena. So these are the examples which comes under whirling movement. So example can be given as euglena or sperms. Coming to location and structure of skeletal muscles, now here we need to understand about various muscles present all over in our body, and hence we come to learn about human muscular system. As you know, all locomotion and movement are movements, but all movements are not locomotion. So this you need to remember what exactly locomotion is. All locomotions, they are movement. Locomotion matlab movement from one place to another, either may be voluntary, may be involuntary. तो locomotion होता है, वो सभी locomotion movement है, but all movements are not locomotion. Like peristalsis movement is not a locomotion. So here we are going to study about what are locomotions here, what muscles comes under it, skeletal muscles as we have studied in one of the lessons, types of muscles, skeletal, where striated or non-striated comes, and even cardiac muscles we have studied. The same we have here again in detail with some pictures to understand a detailed concept about movement in case of muscles. So human muscular system, it consists of, or it includes muscles which are mesodermal in origin. We have studied about 3 different dermal layers: ectoderm, mesoderm, endoderm. So muscles are arising from this particular dermal layer, that is mesodermal layer. Muscles constitute 40 to 50% of the body weight. Muscles have excitability, contractility, extensibility, and elasticity. So these are the properties of muscles, and 3 different types are shown over here. Let us understand one by one each of these muscles in detail. Human muscular system, which firstly includes first type of muscles, those are skeletal muscles. They are attached to skeleton. Striations are present as we have studied earlier. They are voluntary, rich blood supply, fatigue muscles, muscle fiber is multinucleate. Multinucleated hai. More of mitochondria are there in these particular muscles. You can check out how their arrangement is and where are exactly they seen. Next we have visceral or smooth muscles, which are usually seen in the body parts, in the internal organs, which are not moving, which does not show any locomotion. In fact, they are showing movement, but movement like peristalsis and all. These are present in visceral organs, which means the organs which are internally located. Striations are absent here. Involuntary, they have poor blood supply. Non-fatigue, uninucleate, only single nucleus is seen. Less mitochondria are seen in these particular cells. Lastly, we have cardiac muscles. These are found only in heart wall. Striations are present. They are involuntary. Rich blood supply, non-fatigue, uninucleate, and more mitochondria are seen. So this is actually a combination of both striated as well as non-striated ones. And so we could find few features of striated and few of non-striated. Coming to the human muscular system at a glance, this is all the comparison of what we have studied right now, that is skeletal, visceral muscles as well as cardiac muscles, which we have already discussed, but this is a comparison, so note it down or take a screenshot of this particular page. Coming to structure of striated muscle, the details we have studied how the arrangement of the cells are, structure of striated and non-striated or cardiac muscles are. Skeletal muscle or striated muscles, they are made up of many muscle bundles which is called as fascicles. Fascicles are held together by collagenous connective tissue layer, that is fascia. All these was explained already, but this is just a recapitulation of what was being explained so that we can understand the further concepts more clearly. The things are again being given over here. Each fascicle contains many muscle fibers. Muscle fibers are lined by plasma membrane, that is sarcolemma. As you know, sarcolemma, plasma membrane को हम लोग lemma बोलते हैं, या जैसे sarco, this is used for muscles usually. So as sarcoplasm present, that is cytoplasm present inside the muscle cells, is called as sarcoplasm, that is lined by a plasma membrane called as sarcolemma. Next we have here is each muscle fiber contains parallelly arranged myofilaments या myofibrils. Each myofibril has alternate dark and light bands, as we have studied, A band and I band. This is due to the presence of two fibrous contractile proteins, thin actin filament and thick myosin filament. So for understanding the structure of myosin and actin filament, you need to understand where exactly they are present, where are they situated, and for that we need to have a recall of the study which we have done earlier. So actin filament and myosin filament, further we are going to study about those filaments here. Coming back to striated muscles, you can check out where I band is present, A band which contain actin and myosin. I band contains only actin, A band contains actin and myosin, which makes it more darker and is the difference between both of these bands. They are arranged parallel to each other. A band bears a lighter middle region called H band. It is formed of only myosin. A thin dark line, that is M line, runs through the center of ZH zone. So what is M line, what is H band, everything was explained in the last class. I band is bisected by a dense dark band called Z line. Region between two Z line is called sarcomere. Sarcomeres are the structural and functional unit of a muscle. You can check out in detail how the diagram is given about what is I band, what are actin, what is Z line, which is in a zigzag manner, M line, which is actually present in the center, what is myosin, and which is exactly A band and I band. So all the details were given about it earlier also. You can check out this particular diagram which was given already in the text about Z line and M line, and moreover even this also where exactly is I band, H band, H zone and A band. So what is actin and what is myosin, that is also given. They are actually the filaments, the myofibrils which are present over there in the muscles. Coming to some contractile proteins, structure of contractile proteins, that is actin. Let us understand about actin first and then about myosin. Actin filament. An actin filament is made up of two filamentous actins which form double helix. You can check out the yellow diagram given over there, how a double of F actin filamentous actins which form double helix. So जैसे DNA का structure होता है वैसे ही ये approximately similar you can check out over here, which is made up of tropomyosin and troponin. So ऐसे दोनों का helix structure, the three structures which are seen over here is troponin, and the green yes, F-actin is a polymer of monomeric globular G-actins, that is globular actins को G ऐसा नाम से लेटर से इंडिकेट किया है, filamentous actins को F से इंडिकेट किया है. Actin contains two other proteins, namely tropomyosin and troponin, as we have seen right now, the yellow band and even ऐसे rope जैसे red color के lines भी दिख रहे हैं. Two filaments of tropomyosin run along the grooves of F-actin double helix. Troponin has three subunits. तो तीन जो छोटे orange color के दिख रहे हैं, those are troponin, which has three subunits. It is located at regular intervals of tropomyosin. In the resting state, a subunit of troponin masks the binding sites of myosin on the actin filaments. So this is how the troponin as well as tropomyosin, they are arranged accordingly and forms a actin filament, which is actually a complex type of contractile protein. And three different components you need to remember. F actin क्या है उसमें, वो actually एक backbone form कर रहा है actin filament का, वो double stranded protein है. Each strand is composed of polymerized G actin molecules, ये आपको याद रखना है. और one ADP molecule is attached to each G actin molecule. तो जैसे टेक्स्ट बुक में दिया हुआ है, डार्क कलर का ग्रीन कलर का एक दिया है एक मॉलिक्यूल, दैट इज एक्चुअली जी एक्टिन मॉलिक्यूल, जो कि एडीपी मॉलिक्यूल को अटैच्ड है. ट्रोपोमायोसिन ये एक्टिन फिलामेंट है, कंटेन्स टू एडिशनल प्रोटीन स्ट्रैंड दैट आर पॉलीमर्स ऑफ ट्रोपोमायोसिन. तो दो येलो कलर के जो बैंड्स दिख रहे हैं, दे आर कॉल्ड एज ट्रोपोमायोसिन. Each strand is loosely attached on an F-actin. In the resting stage, tropomyosin physically covers the active binding sites of myosin of the actin part. Troponin, it is a complex of three globular proteins. जो three globular proteins दिख रहे हैं यहाँ पर, ये तीन का जो सेट है, that is troponin, do remember, which are attached approximately two-third distance along each tropomyosin molecule. तो येलो कलर के जो मॉलिक्यूल है, इनकी डिस्टेंसेस अबाउट 2/3 डिस्टेंस है, इतनी दूरी पर है. इट हैज एन एफिनिटी फॉर एक्टिन, ट्रोपोमायोसिन एंड कैल्शियम आयंस. द ट्रोपोनिन कॉम्प्लेक्स, ये जो तीन का कॉम्प्लेक्स है, दिस इज बिलीव्ड टू बी अटैच्ड to attach the tropomyosin to the actin. ये उसको अटैच करने में हेल्प करता है. Strong affinity of troponin for calcium ion is believed to initiate the contraction process. तो जैसे मसल्स का मेन रोल है contraction and relaxation, for that, for contractile purpose, for contraction purpose, this particular troponin has a strong affinity for calcium ions, and likewise the contraction process begins or initiates with this particular component itself. Lastly, we have myosin filament, the structure of contractile protein, जिसमें myosin एक प्रोटीन है या फिलामेंट है. Each myosin filament is a polymer of many monomeric proteins called meromyosins. तो meromyosin या monomeric प्रोटीन है, उससे बना हुआ है. एक thick filament है. Check out how the meromyosin has two different parts. One as heavy meromyosin or HMM or cross arm, globular head plus short arm, it projects outwards. तो यहाँ पे head भी दिख रहे हैं, tail भी दिख रहा है. So you will find the head, the region which acts as a head is a globular head, which is actually a short arm called as heavy meromyosin, HMM से denote किया है. और two light chains हैं which are associated with each head. टोटल 4 लाइट चेन्स है, ऐसे 2 इससे अटैच है, 2 उससे अटैच है. तो लाइकवाइज 4 लाइट चेन्स है. लाइट मेरोमायोसिन वी विल कॉल इट एज LMM, ऐसे डिनोट किए जाते हैं. मायोसिन हेड हैज अ स्पेशल एटीपेज एक्टिविटी, इट कैन स्प्लिट ATP टू प्रोड्यूस एनर्जी. तो ग्लोबुलर हेड जो है, दैट इज एक्टिव एटीपेज एंजाइम. इट हैज बाइंडिंग साइट फॉर ATP एंड एक्टिव साइट फॉर एक्टिन. तो ऐसे वो ATP को स्प्लिट करके एनर्जी प्रोड्यूस कर सकता है. मायोसिन कॉन्ट्रीब्यूट्स अबाउट 55% ऑफ मसल प्रोटीन. यू कैन चेक आउट हाउ दे आर एक्चुअली प्रेजेंट. कौन सा आर्म जो है क्रॉस आर्म HMM है, और कौन सा पार्ट जो है वो LMM, मतलब लाइट मेरोमायोसिन ऐसा है. So here we have studied about structure of myosin and actin filament. Further mechanism of muscle contraction we'll be seeing in the next class. So as of now, I'm completing the topic over here. Thank you and have a good day.